Um, Thank you for coming to my presentation. Again, my name is Velisaris. As Ken mentioned, I'm a recent UC Davis graduate student, and I did this project with Project Phoebe at UC Davis, and I did it on how songbird nest predator communities and predation rates change across an urban gradient, using sort of the Black Phoebe as our study bird. So just some quick introductions to our project. This project is led by three graduate students, Sage Madden, Ian Halliburton, and Jacob Johnson, as well as advised by two advisors, Gail Patricelli and Tom Hahn, and is a collaborative effort of a lot of undergraduate volunteer workers. All the data collection and the bulk of the work is made possible through them. So, quick thank you to everybody on this project. And then my role specifically with this project is twofold. So during the field season, I work as a field technician, doing a lot of the data collection that needs to be done, which includes measuring and weighing Black Phoebe nestlings, mist netting and banding, and overall monitoring of nests. And then outside of the field season, I work as a undergraduate researcher. So sort of furthering this independent project that I did, using the data that we collected, and manipulating and cleaning it, as well as this final step here, which is PowerPoint presentation creation and presenting. So just a quick background into Project Phoebe and what we do. Project Phoebe looks at urban environments, and it notices that urban environments sort of have these novel stressors, things like heat, artificial light, and noise, as well as unique predation risks happening in these urban environments. And it looks at urban birds that tend to do really well and thrive in these environments despite these unique stressors. And it wants to see what these urban birds are doing differently from non urban birds that allows them to do so well in these environments. So my project specifically looks at this predation aspect of it and looks at this pattern called the predation paradox. Now, this is something that's seen where in non-urban environments we tend to have decreased predator abundance, while in increased or more urban environments we have increased predator abundance. And it's called the predation paradox because despite this pattern, we tend to see increased predation rates happening in more non-urban environments and relatively decreased predation events or rates happening in more urban environments despite this increase in predator abundance. But this isn't a general rule. It's seen sometimes, but in other sort of studies, it's been found that there is no difference in urban and non-urban environments in terms of the predation rates, or that urban environments do have increased predation rates, as you would expect with increased predator abundance. But this hasn't really been documented well in the Central Valley. So my project primarily wanted to answer sort of what's going on in the Central Valley with respect to this predation paradox. So just some general methods before jumping into the nitty gritty of my project and some important terms to understand. This first one being nest monitoring. So this is how we collect all of our data, and it involves first scouting Black Phoebe nests during the field season. And then once a Phoebe was found nesting, we would check that nest 1 to 2 times per week, and during these checks, we got all of the data that we needed for our project and the undergraduate projects that were happening, which included measuring and weighing nestlings, banding individuals when needed, recording predation events, and anything like that. Another important thing to know is the urbanization gradient, which it is sort of how it sounds, where we have all of our different nest sites on this figure here, and some of them are in urban environments, some of them are in less urban environments, and it's not black and white. We have everything in between. So just when I refer to this urbanization gradient, it basically means all of these different urban levels that we work with, and this is incorporated through the urbanization index. And this is basically just putting a number to the level of urbanization of a nest. Negative values mean it means a nest site is less urban. Positive values mean a site is more urban. And I have two of our different sites here pictured: Russell Ranch, a traditionally less urban site, and Davis, as I'm sure you're familiar with, being a more urban site. And then finally, predator surveys were really important for not just the project, but for my specific project. And these were surveys that were 10-minute point counts of potential Black Phoebe nest predators. They were done or they started as soon as a nest hatched eggs, and then were done every 3 to 5 days until four total surveys were collected. And they took about the period of 2 weeks. So now that that has been said, I can jump into my project and the first part of my project, which is describing predator communities in sort of the local counties across our urbanization gradient. And this was done primarily through these predator surveys that I just mentioned, as they allowed me to see what predators were where and allowed me to sort of calculate relative abundances, so how many predators as well. And after collecting all of that data, I was able to plug it into the ArcGIS mapping software to create this large map. Now, you don't need to look at it too hard. I'll break it down in the next slide, but just some important notes about it before I break it up is that each of our little pie charts here, our circles, represents a different Black Phoebe nest. Each color represents a different Black Phoebe nest predator. And then we also have the shading here where we have these red and purple areas being more urban areas. So breaking this down into these non-urban and more urban sites, I just realized, let me move the Zoom meeting in case that's in the way. But these non-urban sites, things like nest sites along Puda Creek and the Yolo Bypass, seem to have, relative to more urban sites, less predators. We can see between our urban sites, these urban sites being Davis and Sacramento, that the amount of colors in the pie charts are not as many, where in non-urban environments, we tend to see one or two per nest site. But in urban environments, we tend to see more. So in Davis, we have California scrub jays, American crows, and Cooper's hawk. And then in Sacramento, we have all of that and more with things like yellow-billed magpies, brown-headed cowbirds, and house sparrows. So it looks like, at least for this part of the project, the predation paradox holds true, where we have more predator abundance and or diversity happening in these more urban areas. And so quickly, why this might be, it may just be due to a lot of papers denote this due to anthropogenic food sources. So things like dumpsters, feeders, bird feeders help these human subsidized urban predators do really well as their prey items like house sparrows, which are thought of as a very urban bird. It helps them do well, which in turn helps their predators do well. So now, with that being said, moving into the second part of my project, which was testing for differences in the predation rates across the urban gradient. So another really important part of the predation paradox, aside from just predator abundance and diversity, actual what predation events were happening and how much. So just some predictions that I had about this part of the project before I answered it was I thought, contrary to the predator paradox, was that we would see more predation happening in more urban environments. And this was due to higher predator abundance that we saw, as well as observational evidence during the field season. It just seemed like we were seeing more predation events happening in these more urban environments, where in non-urban environments as well, they don't have these sort of novel stressors like heat, noise, and light, as well as it's not a novel environment. If I was a Black Phoebe, I think an urban environment would be pretty intimidating to me. So I thought that they would be better equipped to deal with things like nest predation. So after collecting a lot of the nest monitoring data, as well as the predator survey data, I was able to create eventually this graph. And just to quickly orient us to the graph, we have on the x-axis a level of urbanization. So as I mentioned earlier, we have negative values being less urban sites, positive values being more urban sites. And then on the y-axis, we have this predicted percentage predated, which can just be thought of as a percentage of nests predated given a urbanization index value. So we have two different lines here as well, each one being a different year, and the shades above and below the lines just accounting for error that could exist in the data. So for 2023, for the green line, it looks like as we increase with level of urbanization, so as we get more urban, we have a increased chance of being predated. But it's important to note that we don't actually have enough data for this trend. It's not significant, but it may or may not be there. But for 2024, we do have significant data, and it tells us that as we increase level of urbanization, the opposite is true from 2023, that we decrease the odds of being predated. So just another way of putting this is that in 2024, urban environments have less predation, Phoebe nest predation happening in them, whereas non-urban environments have increased Phoebe nest predation happening. Whereas in 2023, there's no difference that we can say for sure is there. So just to reiterate, the main points of this part of the project is that the predation rates depend on the year. It was different per year. So in 2024, if you're a Black Phoebe and you want to do really well and hopefully fledge a nest, you have better odds of living in a urban environment. Whereas in 2023, it doesn't really matter where you would nest. You have equal odds of being predated. So for the year where we do have significant data, though, 2024, the predation paradox does hold true. And then the last part of this project was just some additional interactions between this predation, urbanization, and Black Phoebes. Some curiosities that I personally had that I wanted to tack onto the project. This first one being or involving California scrub jays. We saw that California scrub jays were pretty mean to Phoebes, and they seem to be involved in a good amount of nest predation events that we were able to witness in the field. So I wondered if the presence of these California scrub jays could predict these differences that we were seeing in the predation rates that I saw. And then additionally, I wondered if Black Phoebe nest predators kind of preferred different developmental stages. So if different developmental stages had different probabilities of being predated, these stages just being eggs, nestlings, and fledglings. Fledglings are Black Phoebes that are 14 days older. So some predictions I had about these were that, as I mentioned, I thought that California scrub jays would, or their presence would drive differences in predation rates just from what we saw in the field and what we thought would happen. And then I didn't think there would be any differences in the chance of being predated at different developmental stages. And so after again using a lot of the nest monitoring data and the predator survey data, I was able to determine that neither were significant. So California scrub jay presence didn't really predict the differences in predation rates that we were seeing. and the predators of Black Phoebes didn't really prefer different developmental stages. Each developmental stage had an equal probability of being predated. So just some quick interpretations on why this might be. For California scrub jays, for that aim, it may just be that the predation of nests involves too many variables. Things like nest defense, parental aggression might be important, as well as we just had pretty coarse data for California scrub jays. We only had data from 2024, and I only used a binary presence and absence. So whether a scrub jay was at a nest or not is what I looked at, but abundance, so how many were there, might be a better predictor of differences in predation rates. And for the developmental aim, it may just be that predators are opportunistic. So whenever they find a nest, they try to predate it. And then it may also be that it takes roughly an equal amount of energy to predate each developmental stage. So the trade-offs might just be the same. So with that being said, thank you for listening. Some quick acknowledgments to my two amazing mentors and Project Phoebe itself, as well as a huge thank you to all of the organizations that make Project Phoebe possible. And I think I will open things up now for questions, and I'll stop sharing my screen as well.
Well, thank you, Quinn. Finn. I'm sorry, my grandson is Quinn. Anyway, very interesting. In terms of the effort expended for various stages of development, can you explain how casing out a nest versus chasing down a Phoebe, an adult, would expend similar energy?
Well, I don't know. I didn't specify well enough, but these predation events that were happening were only on nestlings. So the Phoebes weren't flying yet. They were either in the nest as an egg, as a little baby fledgling, or nestling, or as a fledgling, which is just 14 days or until they fledge.
Okay, thank you.
Yeah,
folks, if you have questions, please put them in the chat. We have a question from Joe Ellen. How was the predation monitored?
So the predation was monitored a couple different ways. I think the first way was through our nest checks. When we checked on a nest, we would observe if it was predated or not, and if it was, we would obviously notice that. But we also set up little cameras on the different nests, and that were motion activated. So we were also able to see as soon as a nest was predated in real time, sort of what was happening, and keep that on file and record that.
Okay. Let's see. Any other questions, folks?
Can you hear me?
Yes.
Okay. My name's Jack Donaldson. I was the gentleman who built all of the Phoebe stands for Eon. I'm just curious, one, if they were successful, and two, how those nesting platforms held up during the season.
I remember him showing them to me, and it was really cool and something that we were really excited about. That was more of something that he did. Oh, well, also one of the graduate students that I mentioned, Sage Madden, is in the chat and said they can answer your question. So maybe I'll just give it to them. They can answer it a little better than I can.
Great, thanks.
Looks like Sage said that they're going to type out the answer to the question in the chat while I answer other questions.
My internet's really bad right now, so I think if I try to answer it, it's going to cut out. So I'll type out an answer.
Sounds like a plan. Any other questions or comments while Sage is typing?
This is Anne. I'll just make one comment to say that and remind, looking at all the names here, a number of you are members of the Yellow Bird Alliance, and Finn's work and several other people on the Project Phoebe received funding from us. And I just feel like it's so worthwhile, and we have that money because you all joined. So I want to say thank you to everyone who's been here. And fun, Finn. Too bad you can't keep following up on this research for years to come.
Yeah. Thank you all. Kevin has a question. Did you collect any data on adult predation?
No, we didn't collect any data on adult predation. We were primarily looking at nestlings, and sort of Phoebe nests themselves. If an adult was predated, I don't think we often saw that. More or less recorded it. We would just sort of see that in their absence more so. But we primarily collected data on the nestlings themselves. We didn't look at adult predation.
Okay. All right. Excuse me. Rose asked, is there any influence of West Nile on corvids and predator abundance and predation rate?
We didn't look at that. That wasn't part of the project, but that is interesting, and something that I'm curious about as well.
Okay. Any other comments? Disconnected. Yeah, she's still on.
I'm still here. It's just a long message, and I'm on my phone because my internet is so bad.
I just wondered, do you have any anecdotal stories that you experienced or could tell about, your time in the field watching the Phoebes?
I think what was a really fun part of the field season for me was that different Phoebe pairs definitely had different personalities, and it was really, really funny. We definitely had favorite pairs. Some were really aggressive and like great parents and would like take turns diving at you and snapping at you, and some really didn't care. And in fact, they seemed to like just kind of watch you, and like it seemed like if you handed them a data collection sheet, they would help. But that was the funnest part, is seeing the differences in the Phoebe pairs and their personalities. But it looks Sage mentioned that they want to jump in, so
go for
yes.
it.
Well, my Wi-Fi disconnected and wiped my entire message, so we're gonna give the talking out loud a try, and hopefully you'll be able to hear me. Can you hear me all right?
Yes.
Cool. Sorry for the technical issues. It's been fun trying to do a day of work with Wi-Fi like this. We did see that the shelves held up super, super well, and they're actually all still up along the American River Parkway in Sacramento. This season, we didn't see the Phoebes use them, but we think that's because we put them up in February after our permits were finalized. And we think the Phoebes do their initial scouting of nest sites in the fall because we saw a lot of them visiting potential nests at that time. And that's not something we knew before starting the project. So basically, we think we might have missed the window where they do their initial scouting of sites. And we've had the nest shelves up all season. They're still up, and we're going to monitor them again next year to see whether the birds use them now that they've been up at the end of the season when the pairs are visiting sites. And we did see some territorial displays near the shelves at the end of the last season. So fingers crossed. We also placed some artificial nest cups on a few of them to see if we could cue the Phoebes that those are like useful nesting sites, because we think they use the presence of old nests when they're deciding where to nest. And I'm happy to send you photos or give you more info on that. You can ask Ian for my contact.
Okay, thank you.
Cool. Glad the Wi-Fi held up.
Yeah, thank you very much. That was good information. I'm glad the nest held up.
Yes, thank you so much.
Yep. So if you ever need more, you know where to get hold of me.
Thank you.
Great. Okay. If there are no more questions, we have our next speaker available, Chris Torres. And Ian, thank you so much. I really appreciate you taking the time to talk to us tonight about your project. And it sounds interesting. It's a good start to your career. Okay. Up next, we have evolution of flamingo smile. Christopher Torres is a bird paleontologist whose research focuses on questions about the evolution of the skull and brain and evolution of bird ecology and what influences extinction. His projects involve studying various groups of birds like flamingos, and relatives, and relatives and rails and early prehistoric birds from the various slices of time in Earth's history. So I really appreciate you folks being here tonight. And Chris, thank you so much for being our speaker. And I will give you the floor, sir.
Sure. Let me just share my screen here. All right. Can you folks see that?
Yes, see your cover slide.
Perfect. All right. Thank you all for having me tonight. I'm very excited to be here. I think one of the very first things that people learn about me when they meet me is how much I love flamingos. And I think for the people who know me, that can get probably pretty old. So I'm really grateful to have a new audience to just dump a bunch of flamingo facts on. So thank you all for letting me come and talk to you guys. So I am an assistant professor, a new assistant professor down here at the University of the Pacific at Stockton. And I am a paleoornithologist. So I study the fossils of birds to try to understand what they were like in the past and how they're different from what they're like now and why they've changed and why some species make it over time and some species don't. And I am an evolutionary morphologist, so I study how the morphologies or the anatomies of birds have changed over time, how their structures, how their shapes have changed, and again, what drives that, and how that reflects changes in the Earth as the Earth has changed. Hopefully, to try to understand why the world is the way that it is now, and potentially how it's going to change in the future. And so my research touches on a bunch of different groups of birds and looking at a bunch of different slices through time, depending on what question I'm interested in at any given moment. So, like Ken mentioned, some of the projects that I work on involve, for example, extinction and survival. So when the world is changing, sometimes catastrophically so. What determines which species make it and which don't? And so I focus on a span of time that spans around 66 million years ago, which was when an asteroid hit the planet and triggered what's called the end Cretaceous mass extinction event. That's what killed all the dinosaurs except for birds. Birds are dinosaurs, so dinosaurs never went extinct, but all the non-bird ones did, and I want to understand why that is, because it's a really depressing fact that if we want to understand how biodiversity is changing in the world today, one of the best comparisons that we have is when an asteroid hit the planet 66 million years ago. And so I focus on a lot of different groups of birds that were around at the time to understand which ones made it and which ones don't, including what's pictured here, which is a close relative of ducks that lived in Antarctica around 68 million years ago, the same time that T-Rex was in North America, but only one of those two lineages made it through that mass extinction event. And I think that's really interesting. And so I want to better understand how that played out. As Ken mentioned, another group I focus on are the paleognaths, the big flightless birds in the world today, ostriches, emus, rheas, things that are often referred to as ratites, as well as tinamous, the only flying member of this group that are spread out across South America. But what really interests me about this group is flight loss. Why would birds lose flight? Being able to fly is incredible. Why would a bird lose that ability? And what's really cool about paleognaths is that every one of those lineages, ostriches, emus, rheas, cassowaries, kiwi, they all lost flight independently. And so I want to know why that is and how changes in anatomy track things like loss of flight. And that interest in loss of flight is also why right now one of the main the main foci of my lab is studying the evolution of rails, especially in the context of flightlessness. There's around 200 recognized species of rails, including both extant and recently extinct rails. And of those approximately 200 species, more than 20 of them have lost flight independently. That's 10% of known species diversity has lost flight independently. And that is fascinating to me. And so my students and I are studying how aspects of the anatomy change in response to flight loss in birds that are actively losing flight. Some of these species lost flight like 3 days ago. And so we're looking at how their anatomy is different from their close relatives who can still fly, hopefully with the goal of better understanding why ostriches and emus and so on lost flight 50, 60 million years ago. Something I think that's really useful to know about a lot of my research is that some of the projects that I work on are questions that have direct relevance to the world about like why the world is the way that it is and how we can better understand how the world is going to change going forward. But I think it's also really useful, especially when there are students around, to talk about how sometimes the things that we research are not as sophisticated as other things. Some of the things we research could be called potentially really dumb. And the only reason that we do it is because we're really interested and curious and we just want to know why something is the way it is. And so my research question when it comes to flamingos and why I do any of the things that I do with them is really simple and straightforward. I want to know what the deal is with flamingos. Why are they like that? Why do they look like that? Why do they do the things that they do? Because they are so weird. And today I'm going to talk about the various ways that they are weird and why it is that I'm so interested to understand where they came from, because they are just the weirdest birds in the world, and that compels me to understand why they are the way that they are. So today I'm going to talk a little bit about just the general biology of flamingos, the things that they do and why they do them. I'll talk about, at that point, I'll jump backwards in time and try to talk about where flamingos came from, at least our current understanding of where flamingos came from. So starting with their earliest fossil record, what came before flamingos were around? And then once flamingos arose, what did the earliest flamingos look like? And how were they different from living flamingos? Because living flamingos have a super iconic appearance. And so how were the earliest flamingos different from what they are like today? When did a recognizable flamingo first appear on the planet? And what did it look like? And how is it different from living flamingos? I'll very briefly talk about how flamingos conquered the world. They spread across the world, and then they very nearly fell into extinction. And currently, they seem to be spreading across the world again, which I think is a really dramatic evolutionary history for a group that people probably did not expect were that dramatic. And then the very last thing I'll mention is some of the is one of the many aspects that students in my lab will be continuing to investigate where all of this came from. But the first thing I want to do is just explain to you why I think flamingos are so interesting beyond the obvious here. So flamingos are probably some of the most iconic birds in the world. That's super easy to visualize a flamingo, even if I wasn't showing you one right now. And that's because they have these really characteristic flamingo ingredients, right? Long legs, long necks, these crazy faces, and of course their really characteristic pink coloration. But everything that makes a flamingo so recognizable is directly related to the way that they feed, which you can see hopefully right here. It might be a little blurry, but hopefully you get an idea of what's going on here. So they are flamingos are filter feeders. They have long legs so that they can wade in the water. They've got long necks so they can drop their heads upside down in the water. Their beak is shaped the way that it is so they can filter food out of the water. And everyone's favorite fact about flamingos is that they get their pink color from what they eat. So flamingos are the most highly specialized filter feeders among birds. No other birds filter their food the way that flamingos do. In fact, if we want to find a good comparison, at least functionally, for what flamingos do when they feed, the best place to look is baleen whales. So flamingo beaks are lined with rows of keratinous lamellae, which you can see right here in cross section. These keratinous lamellae are basically a tiny little filtration mesh. And when the bird closes its mouth but opens it very slightly, those lamellae interlock and create a filter. Their lower beak, which you can see right here, is absolutely massive compared to their upper beak, and it's shaped like a trough that can fill with water, and their tongue acts like a piston, which pumps back and forth, drawing water into their mouths and then pushing it back out, and then their lamellae interlock to form a filtration mesh, which is how they filter their food. And so when a flamingo is walking around, that's what it's doing. If you can see this video clearly enough, you may be able to see that under its jaw, there's a lot of pumping motion. That's its tongue pumping back and forth, pushing water into and out of its beak. Some people may have seen videos or in person what's sometimes called the flamingo dance, where they stand in place and kind of stomp their legs up and down and spin in a circle. What they're doing there, they normally do that in the water. And what they're doing is when they stomp their feet, they are disturbing the floor of whatever body of water they're standing in the sediment at the floor of that body of water, that lake, for example. There's a lot of organics that are just kind of present in that. And so by stomping up and down like that and spinning in a circle, they are disturbing the floor of that lake or whatever, and then they can drop their head down and feed on whatever it is that they are disturbing. So again, this is a super specialized form of feeding that no other bird comes anywhere close to doing. And that is why I think they're so interesting. Flamingos are incredibly highly specialized to filter feeding. And what specialization means in general is being really good at one thing to the exclusion of everything else, which is a really big evolutionary gamble. There is kind of a common wisdom that says the more specialized something is, the more susceptible it probably is to extinction. Because if you're really good at one thing and that one thing goes away, then you are screwed because the thing that you do is gone and you can't do anything else. And so what I'm really fascinated by is, why do birds do that? Why do they become so specialized to the exclusion of other things? Because again, that seems like a really big gamble. And as we will see with flamingos, flamingos have been incredibly highly specialized for an unbelievably long time, longer than you would expect for something that's highly specialized. And so by studying flamingos, I'm hoping to get a better idea of how organisms, birds especially, become specialized in the first place. And what environmental conditions drive that evolutionary change, and potentially what might those evolutionary changes mean for populations, communities of organisms to respond to really catastrophic global change, which is pretty much what we're seeing in the world today. So that is general flamingo biology. Something I think that people tend to underestimate is how much diversity within flamingos there are in the world. So there are actually 6 different species of flamingos. And really quickly, I want to go through what those species are and where they're found in the world, because I think that's really informative in trying to help us understand where flamingos came from, because we're going to come back to this towards the end of the talk. So the American flamingo is the one that probably people picture when they imagine a flamingo. They are the pinkest of the flamingos. They are from all around the Caribbean. Sometimes they're called the Caribbean flamingo, but I think that's a misnomer, because there is this tiny little population of American flamingos in the Galapagos, which you can see right here. That's this little spot of color. Hopefully you can see that. This little spot of color down there under his feet, that's the Galapagos. So there's a population down there. These are by far the most common species to find in American zoos. The second most common is the Chilean flamingo, which is spread all across the southern end of South America. Not quite as pink as the American flamingo, close relative. There are two other species of flamingos present in the high Andes mountains of South America, the Jameses and the Andean flamingos. These are absolutely tiny little guys. They're only present high up in the Altiplano, these high altitude lakes up in the Andes Mountains. They never come down. The Chilean flamingo can go up there, but these guys never come down. They're incredibly difficult to gain access to. And so there was actually a really long span of time in the 1900s where they were thought to be extinct because researchers couldn't find them. It was so hard to get to where they are. Something I think is really fascinating is that even though flamingos in general are all really highly specialized to filter feeding, the ranges of the Chilean, the Jameses, and the Andean flamingos overlap in these high Andean mountains. And all three of these species coexist on the same bodies of water. And even though they're highly specialized, they are subspecialized to the point where you can put one of each of these flamingos next to each other, standing in the same body of water, and they can subspecialize on the depth of food that they're feeding on to the point where they do not compete with each other for resources, which I think is really fascinating. In the old world here, in the Eastern Hemisphere, we've got two very widespread species of flamingos. The greater flamingo, its range is shown here in pink, all across the Mediterranean and down the coast of Africa and on here into southern Asia. And then the lesser flamingo shown here in yellow. The greater and lesser flamingos are often sometimes found in zoos, as is the Chilean flamingo, but the Jameses and the Andean never are. They really are only adapted to living in these high altitude lakes, and they pretty much die the second you try to bring them down and put them into a zoo down here. We're going to get into very briefly later on how flamingos arrived where they are today. They have these really widely dispersed ranges that I think are really interesting. A long time ago now, over a decade ago, some researchers and I, some colleagues, used genetic data to reconstruct the relationships of these 6 living species of flamingos. And we discovered some really interesting things. So we found that these 6 species fall out into these 2 groups of 3 species each. This wasn't entirely a surprise. The relationships here were not entirely a surprise. We can see a characteristic difference in the shapes of their beaks. Here we've got 2 skulls, one representing the bottom group here and one representing the top group. I should point out that anytime you see a scale bar in one of these photos, it's always 1 centimeter to give you an idea of how big or small some of these things are. You may be able to see some really gross differences between the shapes of these flamingo skulls. The key difference is here if you look at their beaks and cross section. You can see that the lower beak of the lower group is much more bulbous than the top the top group. That bulbous beak has to do with buoyancy. So the group at the bottom here tend to have smaller bodies, and they feed from higher in the water column. And so accordingly, their beaks are much more bulbous. They float better. The upper group there are much larger than the other group. Some of them are twice the size of the flamingos at the bottom there. And their beaks are much more compact, which is better suited for feeding lower in the water column. So again, even though they're all specialized in pretty much the same way, they are capable of subspecializing if they need to. What was really surprising is how incredibly young flamingos are. So here, these numbers here correspond to the age of each of these divergences between each of these groups. What I want to point out here is that living flamingos as a group first appeared in the world less than 4.5 million years ago, which means if we are looking at birds at the family level, flamingos are the second youngest family of birds that we have investigated so far. To my knowledge, the only family of birds that is younger seem to be the frigate birds for some reason. It's also possible that there are several songbird families that are younger than these. So often, a lot of these figures don't include songbirds, but in non-songbirds, flamingos are the second youngest group of birds that we've investigated so far, which is something I'm going to come back to because their fossil record stands in stark contrast to that. Something else I think that's really interesting and was a little bit surprising about this recovery is where these birds are located. So the lesser flamingo here is found in all across Africa, but its close relatives are way over in South America. For the longest time, pretty much until we published this study, it was thought that the two species that exist in the Eastern Hemisphere and overlap for a lot of their range must be each other's closest relatives because they are located right next to each other. But actually, each of those two flamingos has a close relative that is on the other side of the Atlantic Ocean. The greater flamingo there, with its range in pink, its close relatives are the American and the Chilean flamingos. And so what this means is that flamingos cross the Atlantic at least twice to achieve their current distributions. But we don't exactly know when that happened, clearly within the past 4.5 million years. But I think that's a really unexpected pattern in terms of their biogeography, in terms of how they got to where they are. And later on, I'm going to share with you some details that I think are even more surprising about this. So really quickly, what came before flamingos? So if we want to understand how flamingos evolved, we need to understand what came before them. So the first thing we need to establish is what are the close relatives of flamingos? And for a long time, based just on their anatomy and their ecology, it was thought that their close relatives were either ducks and geese and anseriforms, because a lot of ducks and geese filter feed, potentially storks, Ciconiiformes, because of their overall proportions, their anatomy, and a lot of people thought potentially Charadriformes, shorebirds, because of their ecologies. So many of them live, this is where the waders are. So many of them live in shallow bodies of water. But when we tested their DNA, their genetics, it turned out all of these guesses were completely wrong. Their closest relatives are grebes, which was a huge surprise when this was first discovered. But subsequent studies using entire genomes have demonstrated that of all of our understandings about how birds are related to each other, flamingos and grebes going together is one of the things we are most confident in. Their entire genomes show that flamingos and grebes are each other's close relatives. But despite that really close genetic similarity, clearly their morphologies are very different. Their anatomies are completely different. We can see that in their skulls. Flamingos have this highly specialized filter feeding skull. Grebes have a range of skull shapes that are all generally long, characterized by having a long, straight beak that's either adapted to a more generalized feeding ecology or a more piscivorous feeding ecology. Clearly, if we look at the rest of their skeleton, there's also differences in specialization. Grebes are very famously specialized towards a swimming ecology, very similar to what we see in loons. For a long time, people thought loons and grebes were each other's close relatives because their bodies are so highly specialized to swimming. If you've ever seen a grebe or a loon try to walk, it's pathetic because their bodies are highly adapted to swimming, not to walking. They spend most of their lives either in the water or in the air, and so they have a really hard time walking. So flamingos and grebes are specialized both in their skulls and in their bodies in very different directions. This group has been called the Mirandornithes, which translates to wonderful birds, which I think is very appropriate because I really like these birds. But this is a really surprising recovery, a really surprising finding in terms of their evolutionary relationships. But what it also does is it helps us better understand what we should expect from an ancestor of both of birds that look either like flamingos or like grebes. So what is the earliest record in the fossil record? What's the earliest trace of a bird that might predate and prefigure flamingos and grebes? So the earliest known fossil of anything that might look like a flamingo or be closely related to a flamingo is this bird called Juncetarsus. What you're looking at here is a reproduction of a nearly complete skeleton of one of these things. It lived in both Europe and North America about 54 million years ago. Just to really quickly point out what's going on here, clearly its legs are right here at the bottom 2/3 of this fossil, and then its skull is up here at the top. This skull is very poorly preserved. if we zoom in on the skull, it's very badly crushed. It's hard to tell a lot of anatomy, even if you're someone who's very highly trained in the cranial anatomy of birds. But what we can see is that its beak, which is shown here on the right, is really pointed. It's really long. It's really straight. That's like what we would expect for a grebe, but not for a flamingo. Meanwhile, the rest of its proportions of the body, long legs, long neck, that's what we expect from a flamingo, not from a grebe. So this thing looks like if you took the body of a flamingo and put the head of a grebe on it, you get this pre-flamingo grebe. What I think potentially is very surprising is the fact that despite us having a nearly complete skeleton of this bird, we know almost nothing about it. And that's because what I'm showing you here is a photograph of a reproduction, a copy, a duplicate of the original skeleton. The original skeleton is currently held in a private collection in Germany by literally a little old lady who hates scientists and so has sworn that no researcher will ever be able to set eyes on this fossil again. It's unclear why she feels that way, but she does. And so the best we have is a really poor replica that really tells us not much at all about its anatomy other than generally what it looked like. So for a long time, until actually 6 years ago now, this was the earliest known record of flamingos and grebes. Just really quickly, I want to point out that some of my colleagues and I published a paper on a handful of tiny itty bitty little scraps of bird bones from Mongolia from about 55 million years ago. So they predate Juncetarsus by about 1 million years. And we found this bone, in that little handful of little scraps. There's this bone which is called the quadrate. If you're unfamiliar with the quadrate, it's a bone that forms the junction of the lower jaw to the skull in pretty much every kind of vertebrate, terrestrial vertebrate, except for mammals. In mammals, that bone that I'm showing you right here has been modified into one of the bones of the ear. But in birds, as well as other reptiles and things like that, it's a bone that's at the hinge of the jaw joint. In life, this fossil is about the size of a dime. It's absolutely tiny. But when an undergraduate researcher, Sarah Hood here, took this bone and compared it to the same bone from every bird she could get her hands on, she found that flamingos were the closest match for this bone, which suggests that this now is the earliest known record of the flamingo grebe lineage, predating the old one by about 50 million years, and also changing the earliest known record from being in North America and Europe or one or the other, we're not sure, now potentially to Asia, which is something I'm going to come back to because that's a very surprising change in how we understand the origins of flamingos. That is currently our understanding of what came before flamingos and grebes. So what happened after flamingos split from grebes? What did the first proto-flamingo look like? So this is where I should point out that sometimes when researchers are using names, the meaning of that name changes depending on whether you're a paleontologist or not. So currently, the order of Phenacopteriformes is the order that includes flamingos. There are 6 living species of flamingos. They're all in the family Phenacopteridae. And so for a neontologist, someone who studies modern birds, though the names Phenacopteriformes and Phenacopteridae mean the same thing. They refer to the same group of 6 species of flamingos. But if you talk to somebody like me, the order Phenacopteriformes refers not only to living flamingos, but also to primitive flamingos, including a family called Paleolodidae. So the Paleolodids were flamingos. They had the same basic proportions of flamingos, but their beaks were super straight. And I'll show you what that looks like. I'll show you a photo of that beak here in a moment. But they were straight-beaked flamingos. Now, today, they are completely extinct, and I'll give you an idea of when that happened here in a moment. But in the past, there was a completely separate family of flamingos that eventually went extinct that had a completely straight beak, which means that they were completely differently adapted compared to living flamingos. And so I want to know, what were they like? Because theoretically, these straight-beaked flamingos were not as highly specialized as filter feeding as modern flamingos. So we need to understand what these straight-beaked flamingos were like if we want to know where the curved beaked flamingos came from. So these straight-beaked flamingos have had a really complicated published history, and I think this is a really good example of the sociology of science and the silly problems that paleontologists in particular run into when we're trying to do our jobs. So this straight-beaked flamingo was first reported in the late 1860s, early 1870s by a French researcher named Milne Edwards. And this guy published this book called The Anatomy and Paleontology of Fossil Birds of France. And in that book, he published just hundreds of fossils that he found, all from basically the same place in France that date back to between 25 and 30 million years ago. And in that big book of fossils that he found a nearly complete postcranial skeleton, a skeleton that did not include the skull, that he realized those proportions look like a flamingo. And so whatever this thing was clearly a primitive flamingo that lived 25 to 30 million years ago. And so he called it Paleolotus ambiguus because he wasn't super positive about that, but it was a flamingo. He also found the tip of a beak that he thought looked like the beak of a crane. And so he called it Problearica problematica, which is a very unimaginative name for something that was clearly giving him a headache when he was trying to figure out what it looked like. The photo I'm showing you right here is a photocopy of a photocopy, which is why it looks so poor, but this is the tip of a beak. And then he also had a nearly complete skull that he assigned to an ibis because he thought this thing looked like an ibis. Only later did we discover that all three of these fossils belonged to the same bird and that that bird was clearly a relative of flamingos. So this photo at the top here is one of only two nearly complete skulls of Paleolotus, of any kind of straight-beaked flamingo at all that we know of. And so here we're looking at the beak from the right side, and here it's being compared to the skull of a living flamingo and of a living grebe. And as you might be able to tell, its beak is much more like the skull of a living grebe than of a living flamingo, despite the fact that it has a body that's much more like that of a living flamingo. I should point out the fact that there's no lower jaw pictured here because we've never found a lower jaw from Paleolotus. We only have a handful of upper beaks, only two nearly complete skulls, but none of them have a lower jaw that we know of. To give you another example of the kinds of problems that we face, someone published a photo of a reconstruction that they built somehow of a Paleolotus, a straight-beaked flamingo, back in 2006. It is unclear what is labeled here as a lower jaw. The lower jaw is pictured in this photograph. It's unclear what that is. It may not even be a lower jaw, let alone the lower jaw to a Paleolotid. For some reason, someone who was building an exhibit at a museum took that thing, glued it to the bottom of a Paleolotus skull, and said, this is a complete Paleolotus skull. So for a long time, there has been this ghost of an image that's floating around the published literature that has led a lot of flamingo researchers astray, trying to understand what the biology of this lower jaw is, when in reality, we don't have a clue. This is not even potentially even a jaw. So this is about all we understand of what these proto-flamingos look like. We don't have enough of its skull to really understand how it was using its skull. We don't fully understand its postcranial anatomy, even though it's got these long legs and a long neck that proportionally look a lot like flamingos. Paleolotids, these straight-beaked flamingos, their legs seem to show some specializations that may be better adapted to swimming than to wading. So it's hypothesized that potentially these straight-beaked flamingos were also swimming flamingos and that they were just on their way to having these crazy proportions that we see in living flamingos. So again, we don't fully understand what these straight-beaked flamingos were like when they were still alive, but trying to understand that, trying to clarify what they were like, is one of the major foci of my lab going forward, and that's something I'm going to touch on here at the end of my talk. So that's what a proto-flamingo looked like, these straight-beaked flamingos. So what about the earliest filter feeding flamingos? If we want to know where filter feeding flamingos came from, where did they come from? If we look at the fossil record, what does the earliest skull, the earliest anatomy of a filter feeding flamingo look like? And how is that different from living flamingos? Potentially that can give us some insight into where living flamingos came from. So what I'm showing you here at the bottom is a line drawing of a fossil of a curved beak flamingo, a clearly filter feeding flamingo that lived in France between 25 to 30 million years ago. This is the same locality that straight-beaked flamingo from France was found, which means that straight-beaked flamingo and this curved-beaked flamingo, this evolutionarily modern flamingo, lived in the same place at the same time on the same body of water. So there's a good chance that they were walking past each other every day on their way to work. So they're from exactly the same time. This bird has been called Phoenicopterus croisetii. More recently, that name has been changed to Harrisinavis croisetii, which I'll point out here in a second. Phoenicopterus is the name of some of three of the six, the genus of three of the six living species. At some point, so this was first described by a Frenchman in the mid 1800s. He read clearly it's a flamingo, and so he said, okay, so clearly this is a part of the living genus of flamingo, so I'll call it Phoenicopterus. But later on, we recognized that this is much older than living flamingos. It shows, it seems to show, based on this drawing of a fossil, it seems to show anatomies that are different from living flamingos. So it cannot possibly be within living flamingos. So it can't be a part of that genus. It's got to be somewhere else. So now it's been changed to Harrisinavis. So in the future, I'm going to be calling this Harrisinavis, just to give you an idea of what's going on here. And that, again, that kind of sociology of science creates problems that we have to disentangle as we look through the literature. Are we looking for Phoenicopterus or Harrisinavis? When depends on who the author of a paper is. It can be really frustrating. So this species and this fossil in particular has a really strange history that I think is really interesting, again, as an example of sociology of science. So this fossil was first reported in 1852. Sometime between 1852 and the 1970s, a replica was made and kept in the museum at France, so the Natural History Museum in Paris. So sometime in this window, we don't know when, but we know that someone made a replica in this window. Sometime between the 1970s and 1984, both the original and the duplicate were lost. No one had any idea where they were. Someone in 1984 went to the museum in Paris, in France, looking for either the original or the duplicate and could not find it. And so they published this paper that said, hey, both of these things are gone. I don't know where they are. We have no idea what this thing looks like. The best we have now is this really poor, difficult to interpret drawing of what that fossil would have looked like. Then in 2012, I visited the British Museum of Natural History in London. And as I was poking through their collection, looking at all of their fossil birds that they had stored up, I was just poking through to see what I could find. And I found this, which is the duplicate that was lost sometime before 1984. This is a duplicate of its original specimen, which you can see down here at the bottom. This duplicate, despite the fact that it clearly demonstrated that, yes, in fact, this fossil did exist at some point, this duplicate is effectively useless scientifically. Anatomically, the details in this are impossible to discern. The original fossil, which you can see down here at the bottom, was clearly broken in half. Someone, when they were making a duplicate, tried to glue it back together, but the two sides don't line up. And in a lot of places, there are things that clearly are bone that are painted. So this entire thing is painted, which is why there's different colors here. Things that very clearly are bone are painted as if they're rock, like everything else. And things that are clearly rock sometimes are painted to look like bone. So this thing has absolutely no scientific value other than to demonstrate that, yes, this thing did exist at some point in the past. So sometime in 2012, after I went from that museum in London, I went to this museum in Lyon, France, because that's where this straight-beaked flamingo skull is held. So I went there to try to look at the skull. And while I'm there, I'm geeking out over the skull because it's incredible. It's one of the most amazing things I've ever held in my hand. And the guy who worked at the museum, who was in charge of giving us access to everything, saw how excited I and my master's advisor at the time, a guy called Marcel Van Tunen, how excited we were to be holding this fossil. And he said, hey, if you think that's cool, I have another fossil that's the same thing. It's more complete. It's better than this one. And no scientist has ever touched it before. Do you want to see it? And I said, dude, what are we doing? Why are we wasting our time with this garbage fossil? Show me the good stuff. And so he goes and he comes back with this. I don't think you need to be a flamingo anatomist to see that these are not the same thing. This upper one, the upper beak is complete. That's the tip of the beak. At the bottom here, it's broken. That fossil is broken. The beak tip is broken. Also, the one at the top here is straight. The one at the bottom here is curved. So very clearly, this thing is a flamingo. It's from the same place that Paleolotus, this straight beak flamingo, is from. But this is not a straight beak flamingo. This is much more similar to a curved beak flamingo. And the second I and my master's advisor saw this, we knew immediately that this is a new specimen of Harrisonavus croisetti, that lost skull. To be clear, this is not the same skull. That first skull remains lost. We still don't know where it is. This is an entirely new one. And so while I was on this trip, after I went to this museum, I saw this fossil, I got super excited, and I knew that this was something I was going to work on. I went to a conference in Vienna, Austria, a conference of just bird anatomists, biologists, paleontologists. And while I was there, I met a woman who was working in the museum in Basel, Switzerland. And her job there was to just go through their records, their collection, and look for interesting stuff. And so I was talking to her and I said, hey, I found this new skull. It's super cool. No one's ever looked at it before. It's amazing. It's got all these cool details. It's this species we haven't seen in person in like 30 years. No one's published on it in a century and a half. It just doesn't have a lower beak or a beak tip. And she said, huh, that's funny. I've been looking through my collection at Basel, and I came across a fossil beak tip and parts of a lower jaw from the same locality that you're talking about, the same age. Clearly, they're a flamingo. Do you want to see them? And I said, dude, what are we even doing? Why are you not showing me these photos right now? And so she showed me these photos. And so what we're looking at here, the blurry bits at the bottom there are bits of the tip of the lower jaw. And then this thing on the right here is the tip of the upper jaw. These are from exactly the same place, the same slice in time. They're clearly from different individuals, but very clearly from the same species. And so we got together and wrote a paper about this where we took CT scans of these skulls and these scully bits, and then we put them back together like a 3D jigsaw puzzle using 3D models to create a reconstruction of what its skull, at least most of it, would have looked like when it was still alive. And so right here, you can see this is a 3D model of the skull. It's color-coded based on different specimens, but they've been pieced back together to try to get an idea of what its skull would have been like when it was complete. And if we compare this to skulls representing the two major groups of living flamingos, what we see is that living flamingos have certain tweaks to this beak that are not present in this early form. The most obvious being both the curvature, it's just not quite as curved as living flamingos, but also living flamingos have this bony blade down the middle of the roof of their mouth that we call a keel. That's where a lot of their filtration mechanism sits on when the keratin is there. That's not really present, or at least it's very flat in Harrisonavus. So Harrisonavus was not as specialized, not as highly adapted as living flamingos, but it was still adapted to the exact same ecology that living flamingos are adapted to, which means flamingos have been doing their incredibly highly specialized flamingo thing for upwards of 25 million years, which is an incredibly long time to be good at exactly one thing and not really anything else. But something might strike out to you if you remember that I mentioned early on that based on genetic data, living flamingos are only about 4.5 million years old, but this thing is about 25 million years old. So how can that be? How can living flamingos, which look almost identical to this thing, be that young if flamingos have been around for that long? And what we find is that living flamingos are relictual. They are a holdover of a group of birds that once was much more diverse in species number, that was far more widespread across the globe, and then they survived a near extinction event. So the last thing I'm going to tell you about very quickly is how flamingos took over the world and then lost their empire and then now seem to be building their way back up. I'm just going to breeze right through this because it's basically a bunch of maps with dots, but some people are geeks about where birds appear and where they don't. And I think it's really interesting. So we're just really going to quickly go through this. So as a reminder, that early flamingo grebe ancestor arose sometime around 55 million years ago, this Tarsus, in Mongolia, and then spread very quickly, probably to Europe and North America. At least this is what the fossil record seems to be indicating so far. But then, if we look at the genetic data that we have that puts flamingos and grebes together, we see that between 40 and 33 million years ago, flamingos and grebes split from each other, but there's a gap where we do not have fossils for either flamingos or grebes. There's this gap in the fossil record where there's nothing. We just don't know where they are or what they look like. Grebes first appear in the fossil record really recently compared to where we are today, and they look pretty much like a modern grebe. Flamingos have a much older fossil record. So the first flamingo to appear in the fossil record are these straight-beaked flamingos that appear in France around 33 million years ago. Almost immediately afterwards, we get these blue stars here in other places in France where they spread out. So each of these represents a species that we know about. This pink dot here represents Harrisonavus. So Harrisonavus first appears almost immediately after these straight-beaked flamingos here in Europe. But then after this, things get really weird, and they start spreading out across the world very, very quickly. We get records of these straight-beaked flamingos here in Egypt. We get a record in Brazil. We have weird trackways in Argentina that people have tried to say are flamingo trackways, but we don't really know for sure. But this thing in Brazil very clearly is a straight-beaked flamingo. So almost immediately after taking over France and nearby European countries, they spread to Africa, they spread to South America, which is largely where we find them today. But also, for whatever reason, they are super diverse and abundant on Australia at this time, both straight-beaked and curved-beaked flamingos. For the record, they are not in Australia today. But between 23 and 33 million years ago, Australia was absolutely covered in flamingos, both curved and straight-beaked. Over the next 15 or so million years, curved-beaked flamingos spread out across the world. They go to Asia. They come down here into southern South America. South America becomes the capital city for flamingos, both straight and curved-beaked. Modern flamingos start to spread out across the world, and we get a straight-beaked flamingo down here in New Zealand, which is kind of interesting. They were in Australia previously. They seem to completely die out in Australia, but straight-beaked ones appear in New Zealand. But we also get a straight-beaked flamingo appearing here in South Dakota. We don't know how it got there. We don't know how many there were. We don't know when it disappeared from South Dakota, but if for at least a few million years, there were straight-beaked flamingos living in South Dakota. Between 5 and 2.5 million years ago, almost all of the straight-beaked flamingos vanish except for one record in Oregon, and that is the last we ever see of a straight-beaked flamingo. The last stronghold of straight-beaked flamingos was the west coast of North America, the United States, and then they're gone. Meanwhile, we have curved-beaked flamingos all throughout North America on both coasts in Florida, in Mexico, up here in California. We find them in La Brea. We find them after this, we find them in the Mojave Desert. So they're all across North America. And they are in one last record is here in Australia about 2.5 million years ago. But these are the only fossils that we know about. If we look at their DNA, we know that some relative of the American greater and the Chilean flamingos, their modern distribution is shown here in pink. We know that that lineage genetically has to have appeared at this point, but we don't have fossils of it, and so we don't exactly know where it is. The lesser flamingo, based on DNA, the lesser flamingo towards the end of the span would already have appeared. Again, we don't have a fossil record of it anywhere, but we know that lesser flamingos were around somewhere in the world, probably somewhere in Africa. Then between 2.5 million years ago and 10,000 years ago, again, for some reason, Australia is covered, in this case, in curved beak flamingos. We get one last record in La Brea and one last record in the Mojave Desert here in California. And then the flamingo fossil record vanishes, and all we have of them are the living species. So flamingos moved all over the world in really strange places. At some point around 5 million years ago, they almost went extinct. All of the straight-beaked went extinct. The curved ones almost went extinct, except for the ancestor of the American, greater, and the Chilean flamingo and the lesser flamingo. And then from there, they diversified and started taking over the world again. And that leads us to today. So the last thing I want to share with you is one of the many directions I'll be taking my research in the future, which is trying to understand what those straight-beaked flamingos were using their beaks to do. And to do that, I'm looking at baby flamingos. So baby flamingos have straight beaks. When they hatch, their beaks are more or less straight. They do not have the curve that adult flamingos need in order to feed themselves, so they can't feed themselves. And instead, baby flamingos rely on their parents to produce what's called a crop milk, which we see something similar. Doves and pigeons evolved something similar independently. And one known species of penguins evolved a crop milk, which is a fluid that's highly nutritious, that's made up of partially digested things that the adult ate, and then it secretes into the beak, into the mouth of its baby. Flamingos use that to feed their babies until they have a curved beak and can feed themselves. Something I think that's really potentially very often disturbing, unless you don't know what it is the fact that that crop milk that they secrete is bright red. And it looks a little bit like blood when they are feeding their offspring. So this red fluid here is that crop milk that the one parent is completely missing, but the other parent is secreting into the mouth of its baby. So this is how adults feed their kids. Baby flamingos are entirely black and white because they can't feed themselves. They don't get the pigments from their diet that they need in order to turn turn pink like the adults. So theoretically, as baby flamingos get older, their beak becomes increasingly curved and they learn to feed themselves, and that's when they turn pink. But sometimes, especially in captivity, that process breaks down. And so sometimes in captivity, we find cases where baby flamingos, juvenile, subadult flamingos, despite the fact that they are fully capable of feeding themselves, they do not stop relying on their parents for food, which I'm going to guess that nobody here can relate to. But what that means is that sometimes in captivity, this is a photo of me at SeaWorld down in San Diego. I was at a conference that involved taking us behind the scenes at the flamingo exhibit. So this is me meeting a juvenile flamingo on what was probably one of the absolute happiest days of my life. And the SeaWorld in San Diego, they have a separate enclosure where they take a juvenile flamingo that will not feed itself until it learns how to feed itself, and then they can reintroduce it back into the general population and it turns pink. That change from having this straight beak into having this curved beak has never been investigated. No one has ever studied how that happens. And so an ongoing project in my lab is going to be trying to characterize this shift in morphology through development from a straight beak to a curved beak in the hopes that that tells us something about how this changed through evolutionary time from a straight beak to a curved beak. So that's all I've got time for you today. I am lucky enough to have 1 billion people to thank for helping me to accomplish all of these things. And if anybody is curious about anything that I had to say, feel free to send me an email. And those other sets of projects that I work on that I described at the beginning of my talk, please consider that a menu of things that maybe you want to hear about at a future meeting. I'm happy to come back and talk about any of those things. But anyway, that's all I've got for you guys. And so at this point, with whatever time I have left, I'm happy to answer any questions that folks might have. Thanks a lot.
Thank you, Chris. Very interesting. I'm looking at the list here of these people you're thanking. I take it your cats gave you a grant, but your dog didn't?
No, it's actually usually the other way around. Our dog is really annoying. He tries his best. We love him, but he's just the worst. So he does his best. He thinks he's helping. He's trying.
Cool. Let's look at the chat, see what we got here. We got thank yous. And Joanne says, I thought I wasn't interested in flamingos. So, folks, you have any questions? What are your questions for Chris? I know there's a lot of stuff, but there must have been something that jumped out at you. Okay, new message. All right. How are you going? Here's a question from Joanne. How are you going to study the transition from straight beak to curved beak in the young?
Really good question. When baby flamingos first hatch, their beak is completely straight, and we know that they go through this really incredible extreme change in their morphology. The bone itself changes shape. To my knowledge, no bird goes through a more extreme change in beak shape after they hatch. I've talked to countless zoo handlers, flamingo handlers at zoos, saying, hey, baby flamingos do this thing. Maybe you've noticed it. I want to know what that's like. And no one's ever written it down. Us researchers have no idea how that happens. Every handler I've ever talked to has said, I could sit down right now and tell you exactly all the various stages that this goes through. So that is going to be our goal, is we're going to try to work with as many flamingo handlers as we can who work with as many of these species as possible to, at a very coarse level, characterize the morphological shifts that the beak goes through as a bunch of different individuals grow up, as they go through these shifts in their morphology. The next step would be to look at CT scans of individuals that die of natural causes throughout this process, to actually characterize what's going on at the bone level, to look at the bones themselves and figure out which bones are changing and how and when and how quickly as they go from being from hatchlings to adults, to try to characterize anatomically what are the shifts that we are seeing.
Okay. So, Joanne, I hope that answered that. Joellen asks, how are young flamingos in captivity taught to feed themselves?
They don't seem to be. So something I think is really interesting is that, so at the very beginning, I showed you that video of various behaviors that are associated with feeding, right? Walking around, dropping their head in the water, pumping their tongue, that flamingo dance that they do as they spin around. Something that we see in baby flamingos as they get older is the fact that juvenile flamingos, baby flamingos that clearly have beaks that are not adapted yet, they're not modified yet to feeding, they go through all of the same behaviors. They show all of the same behaviors that an adult shows, despite the fact that they can't feed themselves. Baby flamingos do that little stompy dance. They drop their heads in the water. They try to move water through their beaks, even though none of it is successful because they can't feed themselves. So by the time baby flamingos have a beak that's capable of filtering, they already know how to do all of it. They just sometimes, for whatever reason, they don't. In more so, as far as we can tell, in captivity, it's not clear what determines which ones are going to feed themselves and which ones aren't. But we know that there seems to be cases where things don't go to plan, and it's not clear to me why that is, or anyone why that is.
Wow, interesting. Okay, Lynette asks, are there any examples you know of where morphological specialization is the ancestral state to a more generalist feeder?
No, not that I know of. That's not to say that it's not possible that it hasn't happened. I just don't know of it. I know of cases where specialization has shifted to a different specialization that potentially is maybe what we see in the early ancestors of flamingos and grebes. It's possible, for example, that the earliest true flamingos, the ancestors of straight-beaked and curved, but not of grebes, was maybe specialized to a more swimming ecology, and then it shifted away from that as it became increasingly invested in filtering. I don't know. But as far as we can tell, we never see that I can think of any examples of going from a specialized ecology, it's certainly not a specialized anatomy, to a more generalist anatomy. It typically seems to be the reverse, going from a generalist to a specialist. But if you know of any examples, I'd love to know about them so that I can investigate them and try to get an idea of why things change.
Okay. Christina asks, what are some of the hypotheses for why flightless birds quit flying?
Probably because they could. Being able to fly isn't great. It's super adaptive, right? It lets you do all of these amazing things, but it is incredibly highly resource hungry, right? The energetic cost of being able to fly is monumental. It's absolutely crazy. It also requires a lot of modifications of the skeleton, what we might call specializations or not true specializations, modifications of the skeleton and the rest of the anatomy to be able to fly. There are all of these constraints on what you're able to evolve to do if being able to fly is something that you need to maintain. The moment you stop being able to fly, you're free to do all of these incredible, amazing things that you couldn't do before. So, really, birds seem to lose flight if they can. And usually that involves being somewhere, arriving somewhere where there are no terrestrial predators, usually no terrestrial mammals, that are going to hunt them or outcompete them. And so we typically see that in really isolated islands like Madagascar, New Zealand, to a degree Australia, or areas that for whatever reason just don't seem to have a lot of terrestrial mammals or large terrestrial carnivores. And so at that point, birds don't really need to fly to avoid predators. And if they're adapted to eating things that they don't need to fly to get to, they don't need to fly anymore and they can give it up and they can use those resources to do literally anything else. So seems to be that they lose their flight if they can.
Wow. Okay. Excuse me. Rose asks, are there any examples in flamingos of changes maintained in populations slash organisms in response to catastrophic global change? If I
Uh,
understood it correct.
Sure. So changes maintained in populations and organisms in response. Yes. So there are. The most common one to see is extinction. That is so in catastrophic global climate change, by definition, um, it is survival and speciation. So something I think that's really important to keep in mind is that the reason why really rapid catastrophic climate change is so catastrophic is because it is climate change that is happening more quickly than species can evolve to adapt to it. And therefore, your options are either hide, do something else without evolving, or go somewhere else or die. Right? Those are really your options. And so the populations, the species that seem able to survive a mass extinction event, a catastrophic change in the environment, are species, especially among birds, that already have, they have pre-adapted, they already have a feature that allows them to survive whatever change is going on. A really classic example is birds in relation to other dinosaurs. Something I'm happy to come back and talk about a lot in the future is research that I and some colleagues have done looking at the evolution of bird brains in comparison to the brains of non-bird dinosaurs. And what we've found is that there are certain modifications of the brain, of the forebrain, especially the cerebral hemispheres, that we don't see in non-bird dinosaurs. And that seems to indicate that going into this mass extinction that happened 66 million years ago, the ancestors of living birds were smarter in some way. We don't fully understand how, were smarter than all other dinosaurs. And so when the world catches on fire and the thing that you're good at doing or the thing that you prefer to do, you can't do anymore, the birds that the dinosaurs that were smart enough to come up with a different solution were the ones that survived. And the ones that couldn't were the ones that died. And so it wasn't the change in environment that caused them to have that modification. They already had it going into that extinction event. So typically, the traces that we see of mass extinction, of catastrophic global climate change, is either extinction, a bunch of things go away all at once, or diversity, speciation, adaptive radiation, which we also see in birds. Because basically, the second that asteroid hit the planet, the ancestors of living birds exploded. Within the first 10 million years, nearly every major lineage of birds that are alive in the world today already existed. They already appeared within 10 million years of all of that niche space freeing up and being available. Birds exploded. Mammals did it after birds. So mammals also exploded in diversity, but not as quickly as birds did. Birds did it almost immediately. And that pattern that we see is another indicator, it's another pattern that we see in response to catastrophic global climate change. So patterns of extinction and patterns of speciation, diversity are really major hallmarks of what we see as the world goes through really massive catastrophes.
Never thought of it that way. Okay. Very, very good answer. Very understandable answer. Okay. So. There's lots of folks talking about how great the talk was. Both you and Fen garnered some fans this evening. And folks asking for you to come back and talk about mass extinction. There's something else someone wanted to talk about. Oh, you mentioned brain evolution.
Yeah, that's closely tied into that.
Like you told me, you've got a whole notebook. You're ready to go. So anyway, I hope everyone enjoyed tonight's program with both Fen and Chris presenting their interests, and just wonderful. And thank you both for spending the time with us.
Thank you guys.
And so, if there are no more questions, I think I'll let everybody go to bed or something. But again, thank you so much for joining us tonight. And thank you, Chris and Fen, for spending the night with us, telling us about your passions. Really appreciate it. Oh, one new question. Is it? Yep. Someone has come back. They said, good deal. All right. Thank everyone, and hope you have the rest of a good evening. Have a good rest of the evening.